Yeah, thank you for that introduction, and I really appreciate the opportunity to come and talk to everybody today. It's really my privilege to be able to come and share some of the research that we've been able to do, but join this great discussion that you've had, national discussion about invasive species, as part of this webinar series. I did look at some of the other talks, wild pigs and grasses, and there's a lot of great stuff going on. And so I'm really happy to be able to contribute to that discussion and share a little bit about this invasive grass that's really a growing concern across North America. And my hope is that by the end of this, you'll have, of course, a little more knowledge about Phragmites australis, and also have some insights into some promising new research areas that are really developing through time. So my name gets to be on the top of that list there, and I only get to talk to you today, but certainly I recognize that there is a great group of people that I'm working with to make this all happen. Many of the names you see on the slide there from my own organization. I'm at the U.S. Geological Survey, so based out of Ann Arbor in Michigan. That's where West Bickford as well. But also partners, collaborative partners at Rutgers University, Jim White, Kate Kingsley, Keith Clay at Tulane. You see Ping Gong and others at the Corps of Engineers in Vicksburg, Mississippi. So what I'm representing is a whole pile of work over a number of years, trying to put it together in a story that's going to be helpful to you. So I'm going to introduce some of these research ideas, and don't have a silver bullet to say that we have or that we have finished products, but we're making good progress, and hopefully I give you some good ideas to be thinking about. Okay, so that should have advanced here. So Phragmites australis, and this is common read, and this plant, as you can see from the video on the left, is quite robust. This is what I'm going to be talking about for most of the next half hour or so. You can see that it grows quite densely. There's a lot of above ground biomass, and based on the picture on the right, it grows very, very tall, very thick, and so very little light gets down to the bottom of underneath the plants where native plants normally would be growing. And it's often found in wetlands, but also very disturbed sites of detention ponds and roadway ditches and disturbed areas. So you saw from the picture how tall and robust it can be, but what you can't see is that underground biomass. And we oftentimes think that nearly 2/3 of the plant is below ground. So that's kind of that middle picture there. You see extensive root and rhizome system that the plant has underground. It's a clonal plant, and so there's a lot of biomass underground. And when we talk about Phragmites, for the most part, we talk about the invasive Phragmites, invasive to North America. And so the image that I have there is from a 2017 paper. Don't focus on the dots, if you can see them, as much as the blob. And so generally the green blob represents the European haplotype, and generally classified as the subspecies australis. But it is important to note that there is a native Phragmites, and that is generally the purple blob, so you can just focus on that. And you recognize that it was all over North America, and it is identified as subspecies americanus at this point. And ecologically, it's different. It's smaller. It's not nearly as aggressive in its growth form. It can be identified pretty well in the field. I'll give you some resources later on for ways to help identify the difference between native and non-native in the field, at least in temperate North America. But the native has these red stems, and it's generally a smaller seed head. And the leaves on the image on the right, the native is on the bottom, non-native is on the right. So there's a little bit of difference in color oftentimes, and then in size and robustness. But overall, its growth form is it plays nice with other plants. It just grows, as part of the natural community that you might see, and doesn't really stick out compared to what the non-native does. So for the most part, when I just default to saying Phragmites, I'm talking about the invasive, especially for this talk. So the invasive Phragmites has lots of impacts. Impacts people, impacts ecosystems in lots of different ways. As you can see from the growth form, it's really hard for a turtle or for wildlife to get through it. Crowds out the native plants, so it can reduce biodiversity. It can impact hydrology, very difficult for water to move through those very dense systems. But it also impacts people. And so the image on the upper right-hand corner, that's actually a 2-story house, and that's a beach. So that's somebody's beach they would like to be recreating on, to get out to one of the Great Lakes, and they're not able to. That's a problem for a lot of folks. Clearly, it changes the aesthetics of systems, but also it could be a real fire hazard, reduce property values, and have other impacts. So it really is an issue across the U.S. and across North America. So here's a little better map in the upper map there. That is from EdMaps, online resource that you can access. It's fairly recent. Green representing reported identifications of the non-native Phragmites, and the bottom one is from the GBIF, gbif.org website. But you can see that this isn't just a U.S. problem or a Canada problem, but it's really a North America problem. And because it has such impacts across the landscape, it's really a high priority for resource managers. It appears as a species in lots of different documents at several different levels of government and organizations. I just grabbed a few here with a Canadian focus, but the Great Lakes Water Quality Agreement, the Great Lakes Commission Strategic Plan, lake-wide action plans, provincial plans, as well as U.S. state and municipal plans, county plans, and so on. So it's a priority, and therefore there's a lot of focus on trying to figure out the best way to manage this plant. So let me just take a pause with that story and introduce this idea of collective impact, because it's really had a big impact in how we have been able to tackle this species and other species in the Great Lakes. So collective impact is a thing. Canny and Kramer in 2011 is one of the first papers to put out there. There have been many papers since, if you look at it, look up papers for it. But essentially, it's this idea that it's a commitment of a group of people towards a common cause, and they work closely together towards that common cause or their common objective. So Canny and Kramer lay out these 5 critical conditions for success. I'm not going to go through them in detail, but I think it's important to know that there's a framework here, and there's a lot of guidance that can be pulled from this if you're looking to adapt this approach and this framework for your invasive species or for another issue. Again, this was initially created for social problems, childhood obesity, these really intractable problems, and has been since we've built off of that. So we'll talk about that shortly. So the idea of collective impact, again, following this framework, you get away from this idea of not only just not bumping into one another as we go about our work and coordinating, collaborating, but to have this continuous reinforcing actions working towards this common goal. So we took this and put this into a paper in 2016 or so. It came out. Heather Braun led that effort, and how it explains how we took this idea of collective impact and modified it for invasive plants, particularly the invasive Phragmites. And through that work and the collaborative work with the Great Lakes Commission, U.S. Geological Survey, and other agencies, we ultimately helped stand up the Great Lakes Phragmites Collaborative. And so the Phragmites Collaborative is this effort that, again, builds on that collective impact framework and really tries to pull together people, science, and action, people, information, and action. And the idea is that this collaborative is facilitating communication among stakeholders and trying to help with that coordination towards the common goal. There's a website there, and this website, greatlakesphragmites.net, there it is again, is kind of the public face of the collaborative, but it's not the collaborative. The collaborative is the organization, the people, and the stakeholders, and the continuous efforts towards this common goal of reducing the impact of Phragmites on the landscape. So if you go to the website, there's a lot of great information there. Even if you're far away from the Great Lakes, I think you will find interesting bits of information and tools and other resources that I think could be really helpful to you related to Phragmites. There's maps, there's newsletters, there's cartoons, there's research updates, lots and lots of webinars. So I encourage you to go check that out as the face of the collaborative. So the collaborative identified priorities and helped kind of consolidate the different ways that people can spend their energy on Phragmites. And one of the early things that came out was looking at the existing control approaches for Phragmites. So the 4 big ones that I think are the 4 big ones here: herbicides, so imazapyr or glyphosate, that pretty common usage in the States, and restrictions and much more difficult to use in Canada. but also people's choices, whether they choose to use those chemicals or not. But it is a tool that's used on the landscape. Mechanically, you can cut it or crush it, burn, or if you have the capability, flooding can be a very good tool as well. These have variable effectiveness. In most of them, if you're targeting Phragmites, they're also impacting other plants and other resources there, so they're not species specific and they're not always effective. And so there's some real challenges with these individual approaches. And so one of the desires that came out of the collaborative was better information on which combinations of these are really best to use, because there are best management guides out there, or best management practice BMP guides, but they're for the most part based on expert opinion and don't have a lot of data to help guide and refine those recommendations. So that helped us stand up this program called PAMF, Phragmites Adaptive Management Framework, as an outshoot of the Phragmites Collaborative. So this is really a system scale effort that is intended to learn from all the individual actions on the landscape. So people do actions to apply some of those management combinations, and then they collect information. They report that information back to a model. The model then makes recommendations back to the user, the manager, and we learn then from the effectiveness. And the monitoring happens again, and so we learn from the process helps us reduce the uncertainty around which treatment works the best given certain circumstances and site conditions. So you can see the website here for PAMF to learn more. It's a voluntary program. There's a lot of great documentation about that if you're in the Great Lakes Basin area, we highly encourage you to check it out and participate. If you're outside the basin, it could be of interest to you to do something similar in your area, or maybe adapt for another purpose. The results I'm not going to get into in detail, but I will encourage you to check out at the website. There's this great dashboard there. Here's a screenshot of it that will show you various metrics of how the program, which is now over 4 years old of operational data, is really starting to reduce that uncertainty. In the upper right-hand corner, you see a map. You'll see those circles represent the number of management units that are active within PAMF. And you notice there's great representation from Ontario, and it's been really fantastic through time to work with everybody, especially in Ontario, in a very collaborative way on this project and this framework and research overall. So I really appreciate those interactions through time. So if we get back to these management tools, we have PAMF going on to try to refine what are the best approaches, or best ways to use these tools we currently have. The message kept coming up, we really need more tools in the toolbox. We really want better ways to manage this. We need more options to apply on the landscape. So that's really kicked off a number of years ago. My two main research themes focused on the microbiome and genetic biocontrol. I'm going to talk about those two in detail, but in the upper left-hand corner there, you could see just representation. I want to make sure that everybody's aware there's a very active insect biocontrol program going on. And Michael McTavish gave a great presentation on that at the Ontario Phragmites Working Group just in January. So there's a webinar out there to get the latest on the work to foster native insects as predators to the plant and use that as a form of control. So that's going on in the US and Canada, testing and research is associated with that. So I'm not going to talk any more about that, but I want to recognize that it is a new method that is being refined through time. So let me first talk about the microbiome a bit and the collaborations that we have and the work that's come out of what we end up calling the Phragmites Symbiosis Collaborative. So I'd like to start with this cartoon just because a lot of people might think, well, like microbes, like bacteria and fungi, like why are you focused on them? Well, there's extensive understanding and continued work to think about the role of microbes in the human body, and we think about the benefits that they confer to us. And there are many more bacterial cells in our human body than other types of cells, and we know that there needs to be this relationship between the microbiome and the human to have good health. And so taking that idea and building on that recognition that we have we have microbes or bacteria on our skin and in our digestive system and on and around us. Plants are the same way. Bacteria, fungi, oomycetes, and other organisms are on and all over in the plants. And we also know that they confer benefits to the plants as well. Drought tolerance, salt tolerance, lots of different lots of different benefits that microbes confer to plants. And I just offer this one paper. This is pretty well known, pretty well published, but this one paper in Science that just lays out some of these ways that microbes help plants function and help them be very competitive. And so our question is, can we disturb that relationship in Phragmites as a form of control? So we started this process a number of years ago, 10 plus years ago, reached out to lots of different folks and trying to really develop this collaborative approach towards this question about defining a microbe-based control for Phragmites. And so we created this, what we call the Phragmites Symbiosis Collaborative, in part driven by the Great Lakes Commission as the backbone agency, which was one of those 5 tenets I talked about of collective impact. We created this institution and has really guided a lot of the research that's gone into learning what microbes are in and around Phragmites and what role they play and how they behave and so on. And so we've been working through that guiding that framework, doing lots of great research, again, collaboratively and building on individual studies. There's been several papers, mostly about inventories of the microbes, because there are advances in sequencing technology and ways to tell what microbes are there, and then tackling that more challenging question of what do they do, what are their roles. And we're not there yet, but the technology continues to change, as does the science. But we're always focused on this target of a microbe-based control for Phragmites. And so again, there's a number of research papers out there. We have this patent application for a bioherbicide we'll talk about a little bit more. But a lot of great work has been moving down this path. And so I'll highlight this paper that we did, we put out in 2015, that took this idea of collective impact, adapted it to Phragmites, and laid out a research agenda. And so that's what this collaborative has been working on through time. And that collaborative led to what's now a close partnership with USGS and Rutgers University to develop a non-toxic bioherbicide. And this is just described on the left there, so you can see the components are all things that we can consume or that we know are very prevalent in our society and how we use. But citric oil or something similar is used to strip that waxy cuticle off the plant. Those of you that work with the plant know that it's a pretty rigid, pretty strong plant and leaf. So the citric oil or something, another stripper used to try to get the bioherbicide, which we're envisioning as a liquid right now. So we're thinking about a spray. Conceptually, think about it like a conventional herbicide. So we need to get it on the plant, and then we need to make sure it sticks to the plant. That's what the glycerol does. And then we have a great paper describing what we call the rhizophagy cycle, but it helps articulate the role of arginine and how that can stimulate the plant to produce ethylene and impact microbial composition. And so we have arginine as an amino acid, have that as a component to this, and sugar, and then yeast, common yeast, to help kind of make it all work and really stimulate the microbial community to help take away some of the nutrients that the plant is able to uptake. So we've been working on this. Did it advance? It didn't advance. Let me back up here. There we go. So our process has been to help work on the composition of this bioherbicide and then test it in the greenhouse. And now we're testing it in the field. And so we've been now starting the summer in 2024, our 3rd field season, to test this non-toxic bioherbicide. So we're doing this on small plots. Of course, lots of replication, lots of different treatment types to really figure out composition, concentration, application frequency and approaches, and then look at the impacts. We apply it and then look at the impact on the plant and then analyze all that, and then make adaptations to move forward. So this is a cyclical process that is based around the growing season. But so far, the results are very, very promising. So here are just a few pictures after a week. We're seeing browning of the leaves are curling. The plant definitely is not responding well to this. A lot of this is desiccation, but very profound impacts in the short term, like within a week. And then these three pictures just represent one plot with the control on the left. This is after the growing season, so this is in September. And so this is one on the left is what our control looked like. And then you see a lot of brown. Brown means it's pretty dead, above ground anyway. And so we're continuing to look at those data and refine the formula as we move this forward. So just here's some of our results and next steps. As I mentioned, we're seeing that it's having a pretty good impact above ground, but we're not seeing as much impact systematically as we would like to see. So we're still trying to tease out the impact below ground and impact on those big rhizomes and the reserves that the plant has underground. So we continue to look into that, as well as looking into the composition of the bioherbicide itself, microbes we're working with, and again, concentrations and so on. So that work is ongoing. And as I mentioned, we're gearing up right now to go out and do further field studies this summer, 2024. And so both that work, the microbiome work, and the non-toxic bioherbicide, and what I'm going to talk about here, the genetic biocontrol, we're not envisioning any of these as the silver bullet. These aren't going to be standalone, the best ever, but we do envision that they're going to have a role. And again, they're going to be in the toolbox to have certain situations that application is going to be useful for folks to at least consider situations. So now let me talk about genetic biocontrol. And for this discussion, I'm focusing on RNA. And so stepping back, thinking about the plant itself and why this approach might be worth our energy to invest in. Think about, why is Phragmites so dominant? Well, as for the pictures in the video I showed, it's really tall. So it can crowd out invasive plants by taking away the light. It has very, very dense stems, so it's hard to get through. Again, crowding out invasive plant or native plants. Grows very, very quickly. That's a rhizome. You see in the upper right-hand corner there. So those are all underground, holding resources. And it has these big seed heads, variable seed viability, but they're still putting out a whole lot of seeds. So as we think about and recognize these different traits, we can also recognize that all these traits have a genetic basis. And so the question is, can we switch off expression of those traits? So that's really the core of what we're trying to do here, is targeting not the genetic code of the plant, but the expression of traits that help it outcompete native plants. And so that really starts with the genomics. And so we've been working with Louisiana State University and Tulane University and others to sequence the genome of the invasive Phragmites. And we published that last year. So there's a paper out there, and in GenBank, the code is all there already for people who are interested in working on invasive Phragmites. But that's really the roadmap. That's now our first understanding of what the genetic code looks like and the genome looks like for the invasive Phragmites. We also are almost done and almost publishing the native genome. That's very, very close, as in we should be within weeks, months, kind of close. So then that will be out there for people to look at. And of course, we're looking at the comparison of the native and non-native to see why they behave so differently and why they are structured so differently. But having those roadmaps really help us identify these key traits that we want to target as potential silencing or limiting the expression of them through time. So as we think about genetic biocontrol, we are talking about this in the context of RNA, focusing on RNA, which is different technologies and different approaches than the DNA of the plant. So by not altering the DNA, we're again just blocking the expression of it, which means that it's transient. There's nothing to pass on to the next generation because the plant is there. It starts to express the traits, and these approaches are targeting the expression of those traits. And so by again targeting specific genes, we're able to limit that competitive advantage that Phragmites has over so many native plants. So that's the idea of what we're working through here. So let me talk about RNA and again how that differs from some of the DNA or synthetic biology technologies that are out there. So some of you have seen this. I've used it for a while, and I just think it's a really good animation. So apologies if you've seen this before, but uh, all plant processes are controlled by the DNA, right? We have the genetic code. And if you recall back to early biology days, that gets transcribed to a single-stranded messenger RNA. That gets translated into a protein. The protein becomes a trait. So this is the process for the plant to express certain traits. So the idea is if we don't want a certain trait, we can just work backwards and target that messenger RNA. So how do we do that? Well, we're building off what Napoli et al. Here first started describing in a paper in 1990. They were looking at petunias and purple petunias, and they said, hey, we can make more money off, I don't know if that's what they said, but they said, hey, we want to make more purple petunias. So they started adding these double-stranded messenger RNAs for make purple code, and the petunias came out white. They're like, wait, what? It should be more purple. Why did it come out white? Well, they really dug into it and sorted out the fact that through this process of gene silencing, it's a natural plant defense mechanism where the plant degrades the messenger RNA related to suspicious double-stranded RNA that's coming in. And so this, again, so there's a natural plant defense mechanism where the plant degrades and responds to the entry of this double-stranded RNA. And this was actually formalized and described as RNA interference and got the Nobel Prize in 2006. And so this is pretty well described in literature. RNA interference, again, is this natural plant process that we are really targeting for a way to target the invasive Phragmites. So I go back to my little cartoon here. If we were to insert a gene silencing agent, a double-stranded RNA for photosynthesize, that's right, let's just keep it generic. If we were able to get that into the plant cells, the plant would kick off this defense mechanism where these dicer proteins would come out, it would degrade that double-stranded RNA, and then all the downstream products, and then we would get a plant that doesn't photosynthesize very well or at all. And so that's the process that we're really working on here, which means that this end product is going to be species specific because we're targeting a really small part of that genetic code. We have the whole genome, we have the whole roadmap out there, so we'll be looking for multiple very small targets that will keep it species specific and then non-heritable. That's back to the transient idea because the DNA never changes. It's just the downstream transcription and creation of the proteins that go to the traits. So conceptually, it is a one-season type of treatment. And so RNA interference has been used for lots of different applications. Again, there's lots of examples out there from human health. It's really developing more ways that RNAi is coming to play as for human health. It's in the food system being tested out. I just used the example here of Arctic apples that are able to keep the apples from browning as quickly using similar technology, but it's also used in the pest world a bit already. So there's already examples, and I just grabbed one here of ways that this technology has already been used for pathogen and pest control. So we're adapting it for invasive plants, and particularly Phragmites at the beginning. European froglet is another species that we are targeting behind. It's a couple steps behind the Phragmites. But it is a high priority species that oftentimes coexists with Phragmites and Great Lakes coastal wetlands and definitely the lower lakes of the Great Lakes. It is a growing concern by resource managers. So we're targeting that species as well. But the idea is that we need to get the gene silencing agents into the plant cells. So we're evaluating these nanocarriers to again penetrate the leaf cuticle to get into the plant cell and all the way into across that cell wall to then have the impact it needs to limit expression of those traits. And this work is primarily with the Corps of Engineers. I should have said that at the beginning at the ERDIC. Engineering Research and Development Center in Vicksburg, Mississippi. And so most of this work is happening out of that lab, but it's all in the lab at this point, and we're seeing good results. We're seeing that there is silencing of gene expression that we're able to measure, with blue being the treatment, that's lower, versus green untreated, that's the controls. But then we really need to see that phenotypic response, and that's with the leaves, the Phragmites leaves in the bottom right there. You see some candidates have a bigger phenotypic response than others do. So we're continuing to work through the vectors used and the right the gene silencing agents or genetic targets that we're finding to be most effective to move the plant, or to move the strategy forward as a potential control for the plants. So again, we're using these nanotechnologies of various forms and using those genomic data to come up with the best overall combinations and testing those on plants. And we're really looking to scale up and hope to be getting things out of the greenhouse, or out of the lab and the greenhouse into the field in 2025 or so. But again, the results so far are very encouraging. I think it's a great opportunity to build off this. We haven't figured out yet, because plants have a lot of defense mechanisms and a lot of things that we don't fully understand. And so we're working through those to get this to a point where we can test out at a larger scale. But the greatest, not the greatest, but like a great thing about this technology and the bioherbicide, but remember the non-toxic bioherbicide I was talking about was not species specific. We're seeing that has pretty general impact on most all plants. But this being species specific and working off the genomic data, we're really anticipating it's not going to be a huge pivot to be able to adapt this technology for other invasive plants. Kudzu in the south for us, or cheatgrass, or lots of other plants that are causing management challenges for folks. We envision that this technology could be adapted for other applications down the road. So that's the way we're thinking about it as we really target Phragmites right now. So with that, those are the highlights that I wanted to share with you, a bit about the plant itself, about our efforts to implement implement this collective impact approach through the Phragmites Collaborative, as well as reduce uncertainty through the adaptive management process of PAMF, the Phragmites Adaptive Management Framework, and also introduce these research themes that we have going on here with the targeted microbiome and targeting the RNAi process. The science is really moving quickly with Phragmites, and some of these are being tested with other plants. And so really stay tuned. I think there's a lot of really great things coming up soon in ways. There'll be new ways for people to manage invasive plants before too long. So with that, I think we have some questions, a Q&A. I think the questions we're going to go into Q&A, and Gabby, you were going to kind of facilitate that. But I really thank you for the opportunity, the invitation to come and talk, and hopefully I provided some things for people to think about.